Yes, please start.
Ladies and gentlemen, esteemed guests and fellow DNA enthusiasts, welcome to the World DNA Day celebration program. We are thrilled to have you all here as we come together to commemorate the marvels of DNA and the groundbreaking discoveries in the field of biotechnology. Every year, we celebrate World DNA Day with great pomp. But for last 2 years, it was not possible to physically arrange the program for pandemic situation and Eid holidays. But this time we have taken different competition in virtually. And you can see the amazing posters and articles submitted.
Nishat, who is another person anchoring with Nishat? Please go on. We may be disconnected from Nishat. From the organizers, please take the responsibility who is with Nishat and go on. Khalid.
Okay, sir. I think Nishat has been disconnected at this moment.
Yeah.
And we have another presenter, but he's, I think she's also disconnected. So this is a problem. So I'd like to say something here.
Yeah, please start.
So, we know DNA is the code of our life. So World DNA Day is an annual observance that takes place on April 25th. It is a day to commemorate the discovery of the double helix structure of DNA by James Watson and Francis Crick in 1953, as well as the completion of the Human Genome Project in 2003. So the discovery of the structure of DNA was a major breakthrough in the field of molecular biology, and it's laid the foundation for the study of genetics and understanding. So I think today we are in a very good celebration, but it could be more better if we have participated in our university in physically, but due to the Eid vacation, it's not possible. So, but this is also important to say that all the participants here is from the outer world and also from our universe in the foreign countries also. There is someone in my friend list. She was from Indonesia also. So I think it's another positive thing, I think. So today I'm going to request Professor Dr. Tofazzal Islam sir to start his talk here.
Thank you, Khalid. And I am extremely happy to be here with all the youngsters, lovers of the DNA research and the DNA all we have the DNA. DNA carries the secret course of all of our life. I am very much thankful to the organizers. I appreciate them for organizing this great event. We have now 95 participants connected virtually from Bangladesh and also from other part of the world. And the organizers all are undergrad students, and some of them are senior students also from Bangabandhu Sheikh Mujibur Rahman Agriculture University, and other supporter outreach activity is supported by Bangladesh Agricultural Olympiad, led by Atikur Rahman Asif. I appreciate all of them. And today we have several segments of in this virtual celebration. First, a keynote speech. I shall deliver the speech, and then we have very distinguished discussants. After my presentation, they will discuss and give more insights about the breakthroughs in DNA research and how the DNA research is important in our life. We have with us Professor Mamun Al Mahtab Shopnil, who is a elected fellow of Bangladesh Academy of Sciences, as well as head of the hepatology department, or possibly Bangabandhu Sheikh Mujibur Medical University. And we have Professor Ahsan Habib from Chemistry Department of University of Dhaka, and also Professor Aparna Islam from BRAC University. We have many distinguished scientists I have seen join from home and abroad. So I welcome all of you, and obviously our students, they will arrange the competition of posters, essays, as well as the quizzes. And at the end of the discussion, the results will be also declared. And it is true, if we have in-person celebration, which we had in the past in 2019, would be fantastic. But anyway, virtual world is now a new reality. I am going to deliver my talk on the breakthroughs in the DNA research and our understanding of the mystery of life. Khalid, is this slide visible now?
Yes, sir.
Okay. And how about the sound? Is it okay?
Yes, sir. Excellent, sir.
Thank you so much. I welcome again all of you to my talk on breakthroughs in the DNA research and our understanding of the mystery of life. And this is really an excellent evening where we shall discuss the giants who contributed significantly to understand the secret course of life. The major breakthroughs will be covered my talk, and obviously this topic is very fascinating. Thousands of people are working all over the world. It is hard to cover all the breakthroughs, but the landmark breakthroughs I shall try to cover. If I miss something, I hope the distinguished panelists and other participants will cover. The story started, in fact, the DNA research gave a big momentum after the discovery of the double helical structure by James Watson and Francis Crick in 1953. And this is the image. They draw the structure and made the structure by using tin and rods, and that paper published in 1953, 25th April, and today is also 25th April. So we remember James Watson and Francis Crick for their discovery, and that discovery actually settled the matter and changed biology forever. Now, what is DNA Day? Already Safa mentioned very nicely, but I would like to briefly tell you that the DNA Day is a holiday celebrated on April 25th in the USA. It commemorates the day in 1953 when James Watson, Francis Crick, Maurice Wilkins, another big scientist on DNA research, Rosalind Franklin, and other colleagues published papers in the weekly Nature on 25th April. The United States DNA Day was first celebrated on April 25th in 2003 by a proclamation of both Senate and House of Representatives. April 25th has since been declared International DNA Day or World DNA Day by several groups. And you know this landmark paper, one-page paper published in Nature. The title was Molecular Structure of Nucleic Acids, a structure for deoxyribonucleic acid. And this paper, I hope all of my students, they received this one as a small gift in my first class, and many of you have added this one. This is not actually the experimental the presentation of the data, but it was more, imaginary, based on the, current understanding. And people believe that the discovery of the crystallographic data generated by the Rosalind Franklin significantly helped to propose this structure, but this structure is still valid for the DNA structure. Today, I shall try to focus the historical timeline and breakthrough in DNA research worldwide and DNA-related research in Bangladesh as well as BSMRAU, and we shall have panel discussion, question answering session, and there will be a quiz after the lecture. And online Google software will be used by our students. So from where we can start the journey to tell the story about the DNA research? I prefer to start it from the Charles Darwin. In 1859, Charles Darwin published his remarkable imaginary as well as the fundamental book, The Origin of Species. Charles Darwin published The Origin of Species, which changes the way of many people view the world forever. And how did he, became the father of the evolution? Because he had a fantastic scientific expedition of 5 years, not did research in the laboratory. He went out through a big ship called Beagles and tried to discover the world. And he found in Galapagos Island some fins, different, morphological traits. And based on his observation throughout the world, he, proposed lots of, theory and in his great work. And at that time, the structure of DNA as well as what is DNA, people didn't know, but his work was very insightful, and he's still known as the father of evolution. Another great scientist who is known as the father of genetics, Gregor Johann Mendel. He discovered the basic principles of genetics. He's the first person to shed light on the way in which characteristics are passed down the generations. He worked on garden peas, and we all know the Gregor Johann Mendel's law, and they are really the base of our understanding about the genetics and inheritance of the characters. So he is known as the father of genetics. The third person I would like to mention, Friedrich Meissner, who identifies nuclei. Very interesting. The Swiss physiologist in 1869, the Friedrich Meissner first identified what he called nucleid in the nuclei of human white blood cell. And now we call the nucleid as deoxyribonucleic acid, the DNA. So he should be remembered by all the DNA researchers for his, fundamental discovery. Next, I would like to mention Sir Archibald Edward Garrod. He's the first person to associate Mendel's theories with a human disease. Mendel's theory, after the proposal of the theory, it was overlooked, but after a very long time, the Garrod, he did experiment and proved the theories in human disease. Next, Professor Albrecht Kossel, who got Nobel Prize in 1910 in physiology or medicine. Why? He gave the chemical descriptions of deoxyribonucleic acids, DNA, and ribonucleic acids, RNA. As we know that the DNA and RNA composed of several nitrogenous bases. For example, DNA consists of adenine, thymine, guanine, cytosine, and RNA adenine, adenine, guanine, cytosine, and uracil. He first, confirmed these 4 nitrogenous bases present in the DNA and RNA. And then Thomas Morgan, he's very, very famous and got Nobel Prize in 1933 for his discovery on heritable information is organized and passed on to the next generation. He did experiment, and his famous flight room at Columbia University produced numerous foundational discovery in the nascent field of genetics. And then in 1944, Oswald Avery identified DNA as the transforming principle. In the 1940s, scientists understanding of the principles of inheritance had moved on considerably. Genes were known to be the discrete unit of heredity. However, it was not until 1944 that deoxyribonucleic acid was identified as the transforming principle, and Oswald Avery is known for that discovery. In 1950, Arvind Chargaff's theory, Chargaff's experiment, discovered that DNA composition is species specific. Very important. Every species contains DNA, but its composition, that means the sequence, varies among the species. And that was a big discovery at that time. And then George Wells Biddle, Edward Laurie Totem, and Joshua Leidenberg in 1958, they got the Nobel Prize for their discovery that DNA sequence contain information required to make proteins. They first proved that DNA sequence of DNA is associated with the synthesis of the protein. Your DNA is a series of nucleotides which, when aligned in a specific pattern, spell out a code. And genetic code, we know that, and this code contains information that instructs cell on how to build proteins and how to function it. That means, in short, their discovery was DNA contains instructions for building life. The Nobel Prize in Physiology or Medicine in 1959 was awarded jointly by Severo Ochoa and Arthur Kornberg. And for which discovery? They discovered the mechanism in the biological synthesis of ribonucleic acid and deoxyribonucleic acid. We all know that in the cell, when cell divides, DNA needs to make its copy to transfer to the daughter cell. So they first confirm how DNA, RNA are synthesized in the cell. And the next very big landmark discovery, I think, the starting point of the discipline of biotechnology and genetic engineering, the discovery of the double helical structure of DNA by Francis Crick and James Watson, Maurice Wilkins, and obviously Rosalind Franklin. Although the three guys, Crick, Watson, and Maurice, they got the Nobel Prize, but Rosalind Franklin, she didn't get the Nobel Prize because she died before the the award of Nobel Prize due to ovarian cancer. It was very unfortunate, but the X-ray crystallographic data, X-shaped data generated by Rosalind Franklin was the fundamental for proposing the double helical structure of DNA. Next, I would like to mention Robert Holley, Hargobind Khorana, and Marshall Nirenberg, who got Nobel Prize in 1968 for their discovery of the DNA sequence is coded. They actually, the code and codon, we are nowadays 3 letters code. We understand very well for the recruitment of particular amino acid in the protein. They first, confirm the codons are the equivalent of word in a sentence. Very interesting. And DNA, if DNA is a textbook. So their discovery is remarkable. And the other person, Alfred Hershey, he got Nobel Prize in 1969 for the proof of DNA as a genetic material of life. And he did lots of experiment and proved it. Another very famous man in the history of biotechnology and DNA research, Frederick Sanger. Sanger sequencing, he got Nobel Prize in chemistry in 1958 for his work on the structure of proteins, especially that of insulin. Insulin, is very, very important protein synthesized in human pancreas, and he elucidated the structure of the insulin, how amino acids are aggregated to make the protein. And through this work, he became the first person to order the amino acids and obtain a protein sequence for which he later won a Nobel Prize. He deduced that if proteins are ordered molecules, then the DNA that makes them must have an order as well. Very, very important. And he got Nobel Prize twice, one in 1958, later in, I shall show you, for his another discovery. And DNA sequence, they must be ordered as protein, in protein amino acids is in order. And then I should mention other 3 guys who got Nobel Prize in chemistry in 1980, Paul Berg, Walter Gilbert, and Frederick Sanger again. And Sanger, at this time, he got Nobel Prize for the determination of base sequences in the nucleic acids. And other 2 guys, Paul Berg and Walter Gilbert, they actually famous for recombinant DNA technology. That technology is significantly impacted in our lives, especially in agriculture. And one of the greatest application of recombinant DNA technology is the GMO insulin production by the bacteria and fungi. So insulin is produced in the human pancreas, and scientists discovered the genes responsible for insulin biosynthesis, and through recombinant DNA technology inserted into bacteria and fungi, and now we can get insulin available worldwide, and worldwide all insulins are GMO, and no country in this world restrict that GMO, because GM insulin saving huge lives, especially the diabetic patient. Another very famous scientist, Barbara McClintock, awarded the Nobel Prize in Physiology or Medicine for her work describing the ability of DNA to move between locations within the genome, which is called jumping genes. That means this is very significant discovery. Her studies using maize introduced the scientific world to a new notion. Small segment of DNA can be moved to other region. Even in the same cell, DNA segment from one region to another region can be moved and change the structure. And these mobile DNA elements are left over from a virus millions of years ago, and they have helped shape our evolution by making extra copies of some genes and breaking others. Really amazing. And in the evolution, the viral contribution to our DNA and other organisms' DNA, as well as the behavior of mobile behavior of DNA segment inside the organism is really evolving and making the diversity. In 1990, the first gene found to be associated with the increased susceptibility of familial breast cancer, breast and ovarian cancer is identified by Mary Claire King. Everyone has 2 copies of both BRCA1 and BRCA2, which are tumor suppressor genes. If a person has 1 altered copy of either gene, it can lead to an accumulation of mutations, which can then lead to the tumor formation like breast cancer. So in 1990, another landmark work by human is the ambitious project of the decoding of the human genome, that is the DNA of human DNA sequence of human has been started in 1990. It was a very ambitious project, and the National Research Council recommended a program to map the human genome, and the human genome was officially started in 1990 with the US Department of Energy and the National Institutes of Health, NIH in USA, and that targeted to complete the sequence and decode all the genes over there by 15 years, but it ended very fast. Kerry Mullis and Michael Smith got Nobel Prize in chemistry. Especially Mullis' discovery was very, very famous and widespread now, the PCR method. Mullis developed a technique known as PCR, and really amazing discovery. And Smith determined how changes in the DNA affect the function of the protein. So their discovery really remarkable, and they got Nobel Prize in 1993. Another scientist, Sir Richard Roberts and Philip S. Sharp, they got the Nobel Prize in medicine for their discovery of split genes, gene splicing. All of the sequence in the DNA is not encode the protein. So in that case, during transcription process, splicing is important and really critical job done by the Roberts and Sharp. And Roberts visited Bangladesh. He's very encouraging guy and still very active and supports the biotechnology very much. And next, I would like to mention another remarkable discovery, that is 1995. Haemophilus influenzae is the first bacteria whole genome was sequenced. And then whole genome sequencing has been started in 1996. A very big decision was taken by the scientists who are involved in Human Genome Project. They met in Bermuda. Bermuda is a British overseas territory in North Atlantic Ocean, and they agreed that genome sequence data should be made freely available in the public domain within 24 hours of generation. That was really a big decision of making science open and help other scientists who are not involved in the project to do further work and to make the result impactful. And you all know that in 1996, animal cloning was successful. Dolly, the sheep, was cloned. And then at 2000, the genetic code of fruit fly. Fruit fly is also one of the important laboratory, organism. Its genome was decoded in 2000, and which has much commonness of the sequence with the human genome. So it complements hugely in understanding the human genome. And the first plant genome, which is known as model plant, Arabidopsis thaliana, was decoded in 2000. And it has many advantages, very small genome size, 135 megabase pairs. Life cycle is very short, and scientists use this plant as a model plant, and some of you may be already been experienced working on Arabidopsis thaliana. In plant biotechnology, this is a very important model plant. In 2002, mouse is the first mammal to have its genome decoded, and mouse has many similarities in their DNA codes with human, and it also complemented to understand better the human genome. You see, in 2003, the Human Genome Project was completed. And although in 2001, the first draft genome, 90% sequence of all 3 billion base pairs were published in as a paper in the journal, but the human genome contains 3 billion base pairs, and it is estimated that total genes present in human genome is 20,000 to 25,000 genes, and very complicated. When it was started, scientists thought that we can get all the secrets of life, but day by day we are realizing that it is not so easy. But people are working now very, very hard to understand the function of all genes in human. And when the human genome was started at that time, sequencing was very expensive. And over the years, sequencing is very dramatically, sequencing cost is reduced. And now using, $1,000, even you can sequence your DNA. And it is very important day by day to have the sequence. In future, maybe all of we have our own sequence, and the personalized medicine and other benefit we will get. Genome sequencing has been the big topic and cover stories of Nature, Science, Cell, and many other journals. And genomics, genome sequencing and genomics of any organism telling us about the ancestry of the human as well as other organisms. And genomics is created as a field of personalized medicine also. I hope Mamun Al Mahtab also share his thought. I would like to remember another guy, Roger Kornberg. His father, Arthur Kornberg, also got the Nobel Prize. And Roger Kornberg received Nobel Prize in chemistry in 2006 for describing and imaging proteins responsible for reading the DNA. This is very interesting information. And next, 2 Nobel laureates, Andrew Fire and Craig Mello, won the Nobel Prize in Physiology and Medicine for their discovery of RNA interference. Very, very RNAi technology, it is a famous gene silencing technology and being practically utilized to make the mutant transgene-free mutants. And then I would like to mention Elizabeth Blackburn, Carol Greider, and Jack Szostak. They won the Nobel Prize for their work describing the structure and maintenance of telomeres, the region of DNA located at the end of chromosomes. telomeres are similar in concept of plastic caps on the ends of shoelaces. They protect the DNA from wear and tear during the process of making a new cell. And telomeres and the proteins that replenish them have since been shown to play critical roles in human health as well as their life expectancy. Then I would like to mention three giant researchers who got Nobel Prize in Chemistry in 2009, Ada Yonath, Thomas Tis, and Venkataraman Ramakrishna. And they did a fantastic job, elucidated the structure and functions of ribosome. Ribosome is the site for protein synthesis, and really very, very critical structure they determine. And Ada Yonath, during COVID, pandemic period, she joined with me in celebration of DNA Day and delivered a fantastic talk. So we remember her. Another interesting finding is that in 2013, European forensic discovered that identical twins have different and distinct, genetic makeup. Before, people believed that before that discovery, identical twins, they have monozygotic identical twins, they have 100% genetical similarities, but it has been proved in 2013 that they are genetically different. That means their DNA sequences are different. And Gordon and Yamanaka got Nobel Prize in physiology or medicine in 2012 for their discovery that mature cells can be reprogrammed to become pluripotent. Very interesting. Stem cell technology. Even it has been now using in Bangladesh as a big technology in our medical science. They discovered that Gordon, in fact, first he worked on adult cell of frog 50 years before the Yamanaka started the job. But at that time, people didn't believe his research result, and he failed to publish the data in good journal, and he stopped that research. But Yamanaka, when he failed to do the surgery, he has a dream to become a surgeon, but he repeatedly failed, and then he was forced to do research, and he started the work from the Gordon work, and 50 years later, he proved that, yes, any adult cell can be reprogrammed to become pluripotent, and if you can, do the genetic modification, he discovered why a particular cell is programmed for proliferation of the, for example, from skin cell to skin tissue only, but it can be reprogrammed and make them pluripotent, and really big discovery. And I'd like to mention another three winners of Nobel Prize in Chemistry, Thomas Lindahl, Paul Modris, and Aziz Shankar. They discovered very important how DNA damage can be repaired, because DNA in our cell or other cells, they are damaged. They are prone to damage by different radiation and other carcinogens. But there is a repair and maintenance system in our cell, and they discovered how nucleotide level as well as base level are repaired. The Nobel Prize in Physiology and Medicine received by two scientists, Ellison and Honju, in 2018. They discovered genes involved in immunity breaks. Our immunity has a break, and if you can overcome the break, then it would be a good therapy against the cancer and other cells, and they discovered the mechanism. And interesting discovery how DNA is moving outside the cell, DNA drive, storing data. Microsoft first booted this one, and it is believed that few milligrams of DNA can be used to store all the data available in this earth. I am about at the end of my talk. And heartiest congratulations to the winners of Nobel Prize in Chemistry. I think the most remarkable discovery made by Emmanuel Charpentier and Jennifer Doudna. They discovered the sharpest, technology called CRISPR-Cas genome editing. By using that technology, we can edit the DNA of an organism like Photoshop or Microsoft Word processing. We edit the text or like that. And they discovered the technology in 2012, and they got the Nobel Prize in 2020, within 8 years after their fundamental discovery. And in last 10 years, their discoveries have been utilized by almost all organisms. Any organisms can be edited. Here you can see in the left-hand side of what have been done. Many crops genome have been edited now in the field. In Japan, gamma aminobutyric acid enriched tomato and fish like kingfish, red sea bream with high muscle production. Pufferfish, 3 products are now in the Japanese market, soybean and browning, resistant mushroom in American market, and gene editing has already been utilized to cure the cancer. Alisa, one girl, suffered from blood cancer in UK, already been, base editing helped to cure her. And lots of, multiple, gene at a time you can edit, and next 10 years, it is expected that the gene editing would be a powerful revolutionary technology for both industrial, fourth industrial revolution as well as fourth agricultural revolution. And DNA RNA-based technology also helped us to fight against the COVID-19 pandemic. RT-PCR obviously targeted the nucleic acid, and obviously genome sequencing of SARS-CoV-2 virus helped us to monitoring and surveillance. And vaccine, all vaccines are recombinant. Vaccine, RNA vaccine or DNA vaccine, they have been utilized. So DNA research is hugely been impacted in our health as well as our food security. I'd like to mention 2 other guys who discovered how we can sense the temperature and touch. And they discovered the receptor and expression of the particular gene in the DNA. The last slide is in 2020, Sevante Babu. He got the Nobel Prize in Physiology and Medicine for his discoveries concerning the genomes of extinct hominins and human evolution, especially Neanderthal as well as Denisovan, which our hominins have been extinct a million years ago. And Pabu confirmed that by using the those DNA, damaged DNA. He developed the method and compared with our sequence, Neanderthal and Denisovan sequence, and it has been found that Neanderthal and Denisovan sequence, about 2 to 6% of our DNA sequence are from those 2 hominins. And more interestingly, those who have Neanderthals sequence, especially Europeans, they are prone to COVID-19 disease more than us. Really exciting discovery. And in Bangladesh, Bangladesh utilized the recombinant DNA technology and first introduced the GM food crop BT brinjal in 2014 in the farmers field, and it hugely impacted in the brinjal production. And Bangladesh rice research, Bangladesh agriculture research is highly credited for that. And golden rice have been developed by Bangladesh Rice Research Institute, and it is now on the process of release. And Philippines already released the golden rice. And golden rice contains vitamin A in addition to carbohydrate and other nutrients. DNA research in Bangladesh, if we can summarize, in Bangladesh, the whole genomes of jute, hillsha, goat, year-round jackfruit, and many pathogenic microorganisms, including cholera pathogen, Vibrio cholerae, or SARS-CoV-2, done by many Bangladeshi researchers. In our institute, we use the field pathogenomics to determine the genetic identity and origin of the wheat blast in 2016, which devastated 15,000 hectares of our wheat. And we also utilize the DNA research to discover two genome-specific primer and utilize CRISPR-Cas technology for developing a rapid diagnostic protocol for detection of wheat blast fungus. And we also sequenced many probiotic bacteria as well as utilize CRISPR-Cas technology for editing the genome of wheat for development of blast-resistant variety. And we all know that in 2016, a devastating wheat blast fungus destroyed 15,000 hectares of wheat. And we estimated that it was about, 20,000 crores taka was crops were lost and yield loss up to 100% in some cases. We used the RNA sequencing analysis and determine the origin of the fungus from where it came to Bangladesh very precisely with global scientific community. And that paper we published in BMC Biology, which is the highest cited paper in the history of wheat blast. And that paper helped us a lot to decide the policy decision because when you understand the genetic identity of the organism by genome sequencing, then you can take the measures. In our case, our policymakers took proper decision and tackle the future epidemics in Bangladesh. We decided to discover a precise molecular detection tool because wheat blast was spreading not only Bangladesh, it moved to Africa. And by sequencing the fungus, we found two, nucleic acid segment in the DNA of Magna Portavariae triticum, the pathogen of wheat blast, which is unique and cannot find any other sequence. And that two sequence we utilized for making the primer as well as use the Cas12a CRISPR-Cas technology to make a smart rapid diagnostic technique. Anyone within 30 minutes without any technical know-how, you can identify these. We have made this technology the finest one, not only wheat blast, any disease. If you know the specific unique nucleic acid fragment, even RNA or DNA, you can utilize this one. And Bill and Melinda Gates Foundation recently offered us $7.5 million funding to utilize and apply this technology to 22 countries. So this is one of the greatest innovation in the history of agriculture. And wheat has a very complex genome. You see, it is 40 times higher than the rice. And in 2018, whole genome was annotated. And we tried to understand how can we tackle the wheat blast by utilizing global knowledge and experience of global scientific community. We recently published a paper in PLOS Biology, and we analyzed the DNA of the fungus as well as the wheat. And we pinpoint that both, wheat blast in Zambia, Africa, as well as Asia came from South America, and they are clonal lineage. And we also discovered that RMG8 gene in the wheat genome can be deployed for developing the blast-resistant variety. And this is a recent discovery, April 11th, it was published, and there are 34 authors and 8 from our team. And it was also in many news outlets, including Weekly Nature. our press release was published. And then we tried to utilize CRISPR-Cas technology because wheat is one of the important major food crop. And Sufyan, he is the first guy who utilized CRISPR technology in 2013, after 1 year after the discovery by Jennifer and Emmanuel in the plant. And we invited, he's a Royal Society Fellow and Nick also Royal Society Fellow. They trained us and we edited 10 S genes, negative regulators in the genome of wheat. And finally, we developed, wheat blast resistant, moderately resistant plant. And our idea received the, Islamic Development Bank award. And we also sequenced many probiotic bacteria that can be used as alternative or supplementary to the synthetic chemicals and fertilizers, and discovered the genes responsible for their mechanism. Interestingly, during COVID-19, we did some work. We had a question, why severity of the SARS-CoV-2 infection is weaker in Bangladesh? We analyzed the microbiome in the nasopharyngeal tract of human, healthy, infected, after recovered, and we concluded that SARS-CoV-2 infection reduces human nasopharyngeal commensal microbiome with inclusion of pathobiomes. The dynamics we studied, and very interestingly, Bangladeshi human nasopharyngeal tract contains beta coronavirus, which occupied the SE2 receptor. The origin we don't know, and it may be one of the reasons of lower infection of SARS-CoV-2 by in Bangladeshi patients. And Dr. Nazmul, he is a very powerful metagenomic expert. We published several papers on our research, including how initially epithelial cell in the nasopharyngeal tract respond to the SARS-CoV-2 viruses. And our recent discovery, the whole genome sequencing of a year-round fruiting jackfruit, we decoded it, and our Honorable Prime Minister, she appreciated it. And because the variety Barikatal tree, which was originated in the Khagrasuri forest, Rambor, that variety gives fruit, a fourfold higher fruit than our annual, and it gives fruit from September to June. Really amazing if we can utilize the genomic technology to make new varieties and commercialize jackfruit. That would be fantastic, because Bangladesh is the second highest jackfruit producing country. Every year we produce huge 1 million tons of jackfruit. I always advocate open science to make science for all, and this is the time for making the slogan and to share the data. Lastly, I would like to remember the quote of a Nobel laureate, Norman Borlaug, who made the statement that change we must or we will perish as species, just as did the dinosaur in the late Cretaceous. And we have an Illumina sequencing platform. Who are interested, please welcome. And thank you very much for listening my long lecture. And I hope it would generate some discussion from you. So you are welcome discuss. And now one by one, I expect our organizer will announce the discussant. 3 discussants are now here. Dr. Mamun Al Mahtab, Aparna Islam, and Professor Ahsan Habib, 3 joint professors from Dhaka University, Bangabandhu Medical University, and Prague University. And after that, we may have more questions and answers from other participants. Thank you so much. And now, Nishat, it is your turn. You can invite discussant for their comments and their discussion. Thank you.
Thank you, sir, for giving such a beautiful speech. Speech was captivating and engaging, and it left our audience with a deeper understanding of the importance of DNA in our lives. And sir's ability to explain complex concepts in a clear and concise manner was truly impressive. In fact, we listened to sir's speech as enchanted. It's really beautiful. Thank you so much, sir, again. So, World DNA Day is an annual global celebration of the discovery of DNA double helix structure. This year is extra special. People across the world on April 25, 2023 will commemorate both the 20th anniversary of the Human Genome Project's completion and the 17th anniversary of the discovery of DNA double helix. Today, we have the distinct honor of welcoming our one of panel discussant, Professor Dr. Mamun Al Mahtab Shopnin Sir, Head Interventional Hepatology Division, Bangabandhu Sheikh Mujib Medical University, and Fellow Bangladesh Academy of Science, who is a renowned expert in the field of genetics and medical science and has made significant contribution to the world of science. I would like to request Sir to say something in our program.
You are muted.
স্যার একটু আনমিউট করেন কাইন্ডলি স্যার।
Sorry. Firstly, I must thank the organizers for inviting me to join this discussion as a panelist. It's a very important lecture on a very important day by a very important person who's a renowned scientist of the country. And as always, we have enjoyed the talk by Professor Tofazzal Islam, who has actually covered all the different aspects, right from the very beginning to the most recent progress with the vaccines and the current research with golden rice, etc., that is going on here. So I am a physician, and in clinical practice, although we are not in that sense medical scientists, but in clinical practice we come across the DNA every day. Because there are not only because we are humans and we are living beings, so DNA is a must in our body, but also because of the fact that DNA has become very important in diagnosing diseases. For example, I'm a liver disease specialist, and in our everyday practice, we deal with patients with hepatitis B virus and hepatitis C virus. Hepatitis B virus is a DNA virus, and without testing the HBV DNA or hepatitis B virus DNA, we cannot treat the patient. So DNA is a part and parcel of our life. And nowadays, the application of DNA in everyday clinical practice is increasing. For example, we had known for long that we can identify organisms by culture and sensitivity tests. We do the culture and do the sensitivity to find out which antibiotic works or not, which takes usually 3-4 days to get the result. And also we cannot test for all bacteria that is known to us. But nowadays the DNA sequencing has come, and within few hours, we may know what infection is causing the disease. And DNA, these are only two small examples that where DNA is very important in our clinical practice. But DNA is also the basis that is helping us to expand the research horizon. If we think about research in medical field, the reality of life is that we are only involved with clinical research, but research does not end here. Research starts from the bench, and then it comes to our bedside. But again, if you want to liaise with the bench and the bedside, make a connection between the bench and the bedside, then again DNA is there. For example, to cite another example, Professor Tofazzal Islam was talking about probiotics. This probiotic is very important. For example, now we are thinking of treating many conditions like liver disease, fatty liver with probiotics. But if you want to do a probiotic therapy, you must know the gut microbiota. How is the microbiota of a normal individual in Bangladesh? What is the microbiota of the gut in liver cirrhosis patients, in fatty liver patients? Second, DNA comes into play. So I think that DNA is actually the basis of modern scientific research with a lot of clinical applications. And I think that it is a very useful platform and a very useful day, because if you want to take our science to the world standard, the basis is DNA. What I always say is that we are, বাংলায় যেটা বলা যায় যে আমরা তো চিকিৎসক, আমরা চিকিৎসা বিজ্ঞানী নই, কিন্তু চিকিৎসা বিজ্ঞানকে যদি এগিয়ে নিতে হয় তাহলে চিকিৎসকদেরকেও বিজ্ঞানী হতে হবে, এবং চিকিৎসককে যদি বিজ্ঞানী হতে হয় তাহলে চিকিৎসকের সাথে বিজ্ঞানের যোগাযোগটা স্থাপন করতে হবে। এবং সেই জায়গাটাকে ডিএনএ দিয়ে স্থাপন করা সম্ভব। Like Professor Tofazzal Islam and I can work together if we believe in DNA, and DNA makes the bridge. So that is the concept. So if you want to take our science forwards to the next level, that is very important. Once again, I thank the organizers for asking me to be here as a panelist. I was very excited about the day, and I have requested Professor Tofazzal Islam that this celebration is virtual this year because of Eid and other things. But next year we will also join, hopefully, the physical celebration of DNA Day under your leadership. Thank you very much.
Thank you so much, Professor Dr. Mamun Al Mahtab Shopnil sir, for his informative speech. Now, I would like to give floor to my co-host, Khondokar Afsara Tasnim Totini.
Thank you so much, sir, for your very informative speech. I am Khondokar Afsara Tasnim Totini, hosting on this program, DNA Fest. In our DNA Fest, we have another galaxy of intellectuals with us. Dr. Muhammad Ahsan Habib, sir, professor from University of Dhaka, Department of Chemistry. Dr. Ahsan Habib, sir, earned his MPhil degree from Bangladesh University of Engineering and Technology. Dr. Ahsan Habib has 57 papers published in major journals and has spoken of numerous international conferences. Now, I would like to invite Dr. Ahsan Habib, sir, to take a floor and to share your speech with us. Thank you so much, sir.
Sorry, actually, my camera is not working.
It's okay, sir. You can continue.
Maybe I can, but my mobile is okay. I can continue. Actually, thanks to Dr. Tofazzal Islam for your very excellent work, excellent talk. Actually, I started my DNA work during my PhD in Japan, though I belong to chemistry, fundamental chemistry. Professor Tofazzal Islam, he pointed out the history of DNA, the discovery of DNA, the sequencing, and the latest information that already Dr. Mamun already mentioned. After this talk, I got some fascination in working with actually Professor Tofazzal Islam, particularly in DNA chemistry or DNA science. My work on DNA basically fundamental, something like the free radical DNA cleavage, what the chemicals we are using in our laboratory, in chemistry and other laboratories. We are using many chemicals, but we don't know the in situ effect of the chemicals. A particular chemical may not harmful, may not toxic, but when it combines or they combine with others, they may toxic. So it was my motivation during my PhD, and I started the work on DNA during my PhD. Still, I am continuing, but as you know, we are Bangladeshi. We belong to our educational or other institutional affiliation. The work, the good research, actually we need to do good research, but unfortunately, the atmosphere not so favorable for us. So I would like to request Professor Tofazzal Islam, if it is possible to make a platform nationally to establish, not university level, maybe it is very difficult, next 50 years, it is very difficult to do good work. So that is why the alternative should be not the Atomic Energy Commission, not the science lab or other research institute. There should be a very free research institute like in Japan, in Tsukuba, they have the science city. The science city belongs to the food chemistry, food science, the forestry, all branches. Initially, actually, it is very needed to establish or to make a platform to organize the people. Actually, in Bangladesh, there are many good people, good researchers, good professors. They want to contribute for the nation. Unfortunately, the atmosphere is not favorable. So my very specific proposal to establish is center of excellence or something like the science city, and belong that science city, there should be very major, initially should be major wings, research wings, then gradually it would be flourished. And that research wing should be belong to Prime Minister office. Prime Minister office directly handle that office, not via the bureaucrat. So the fact is the scenario at university level, maybe hardly 10% professors they are engaged with research or good research. Is it possible this 10% or 12% university professor can contribute, can change the scientific atmosphere in Bangladesh or at our university levels? Impossible, not possible. And actually, during my JSPS at University of Tokyo, I was fascinated to work on homochirality, the origin of life. As we know, in our human body or living body, the DNA is dextrose. This is chiral, but dextrose, there is no pair of DNA. The pair is the levels, the protein. So how we can pay our attention at initial stage in Bangladesh, the origin of life, to study homochirality. So finally, my heartfelt thanks to Professor Tofazzal Islam to invite me. Actually, really excellent event. So I again thanks to Professor Tofazzal Islam to invite me in this seminar. And next, in future, maybe we can share our views, our thought, and do something new. Thank you so much.
Thank you so much, sir, for being present with us and for sharing such wonderful knowledge with us. Dear audience, if the question arises, what is the secret of life and what is the source of all of our actions and what governs of all of our expressions, the answer will be DNA. Dear audience, another guest of honor wanted to attend in our fest, Dr. Aparna Islam, but we regret to inform you that she couldn't join with us due to some issues. Dear audience, now I would like to the floor to my co-host Nisha Tasnim Safa.
Thank you so much for Khandukar Afsara Tasnim Totini. We listened to the speeches of our guests. Now we organized a quiz competition based on their lectures. Those who are interested can participate, and there are attractive rewards for them. We are giving the link in the chat box. Everyone, please check the chat box.
Nishat, please open the floor for question and comments from the participants, because we have still more than 100 participants. And when I was talking, I saw around 140 or more participants. So let us hear from the other participants. I saw many distinguished participants from home and abroad. They are joining here. So please allow them and welcome them to ask question or make a short comment.
Okay, sir. Thank you so much. If any question, feel free to ask our honorable guest, Professor Dr. Tofazzal Islam, sir.
Me or Professor Mamun or Professor Ahsan Habib, anyone we can answer. Please check, someone already raised the hand and announce the name and allow to ask the question or make comments.
I would like to request Dr. Mohammad Mainul Islam, sir, to create the question to our honorable guest.
Thank you for giving me the floor to make some comments on the presentation. Very excellent presentation. It was exciting when I was listening and here observing the presentation, and it was very informative and knowledge reachable presentation. And but when I was listening, I was the time passed very fast, but I was looking for more slide. But at the very fast, the time goes very fast. Actually, I have a request, Dr. Tofazzal Islam sir. There are a lot of scientists and teachers working around the Bangladesh. Many they are working on DNA, like many scientists working on microbial DNA, plant DNA, human animal DNA research, many. But when we search in there is no compilation, there is no workshop or book, paper, or any where we can find all this information. Research going on in Bangladesh, the information is very lacking because there is no focusing on compilation of all this work going on in our country. So if our Tofazzal Islam sir or other pioneer researcher take initiative to organize any workshop or publish any book, what research on DNA is going on in different university and institution, then we will get a very valuable book or papers what we have done already in Bangladesh. Thank you, sir.
Excellent. Dr. Mainul, he did PhD in USA, and currently he's leading a laboratory, ISO certified laboratory for health certification of plants, and he recently joined our group as a postdoctoral fellow, and he will work with University of Saskatchewan and Global Institute for Food Security. Thank you, Dr. Mainul, for making this comment. Yes, Dr. Mamun Al Mahtab and Ahsan Habib and many other, Professor Samsur Rahman, many people are working on DNA research. We may take an initiative to compile a book in last 50 years in the history of Bangladesh, how much work we did, all the breakthroughs, including the breakthrough of Dr. Farooq, who first elucidated the evolutionary biology of cholera pathogen, was the cover story of Nature. Yes, we have some outstanding work scatteredly being done in many researchers. And if we can compile all, that would be very nice. And Dr. Ahsan Habib also made some comments, how can we go on? Obviously, we need to work together and focus, which we have done so far, and make a plan. I hope Dr. Mamun Al Mahtab and other professors, Professor Ahsan Habib, Shamsur Rahman, Professor Noman Farooq, we can may try to arrange a national program in next DNA Day and invite all the researchers who did significant work on DNA research in Bangladesh. And obviously, we can invite some guests from abroad. Bangladesh Academy of Science can be also involved. Thank you so much, Dr. Mainul. And I am expecting more comment and suggestion or even a specific question.
Okay, yeah, Professor Tofazzal Islam. Can we do something like to publish a souvenir, and we can invite the professor or researcher? They can provide some writings, and we can compile.
Yeah, this is a good idea. I think Dr. Swapnil also made a comment.
Sorry, not like the full research, but the concept, the thought.
I understood the point. One of the point many people suggested me in last 50 years in fundamental discoveries, what were our accomplishments? There should be a book, covering all the disciplines in the global mass of knowledge, which how much we really contributed. There should be a like a class of scientific discoveries by Bangladeshi researchers. Yeah. We can do it. And your point, Professor Ahsan Habib, is also interesting. We may make another meeting after this Eid vacation. All of us are now in holiday mode. Dr. Mamun Al Mahtab, do you have any comment on this point discussion?
Thank you, Professor Tofazzal. আমি যেটা আসলে বলার চেষ্টা করছিলাম যে বাংলাদেশে মানে আমরা তো আসলে একটু পিছিয়ে আছি আমরা বিজ্ঞানের দিক থেকে চিকিৎসকরা। তো আমার কাছে এটা মনে হয় যে আমরা সবসময় বিজ্ঞান নিয়ে কথা বলতে গেলে যেটা মনে করি যে আমার সেন্টারে কেন ফেসিলিটি নাই, আমার ল্যাবটা কেন আপ টু দ্য মার্ক না। আমার কাছে মনে হয় যে আমি যেটা ঘুরে ঘুরে আমার এক্সপেরিয়েন্স যে বাংলাদেশের বিভিন্ন প্রাইভেট এবং পাবলিক ইউনিভার্সিটি ইনর্মাস রিসোর্স লুকিয়ে আছে। আমাদের যেটা সমস্যা বা সংকীর্ণতা বা ব্যর্থতা সেটা হচ্ছে যে আমরা এক বিশ্ববিদ্যালয়ের এক ডিপার্টমেন্ট, পাশের ডিপার্টমেন্টে কি হচ্ছে সেটা জানি না, এক বিশ্ববিদ্যালয় আরেক বিশ্ববিদ্যালয়ের আমরা জানি না। আমি বঙ্গবন্ধু শেখ মুজিব মেডিকেল বিশ্ববিদ্যালয় আছে, আমি ঢাকা ভার্সিটি কিছুই জানি না। কিন্তু যদি আমরা একটু রিচ আউট করার চেষ্টা করি, আমার কাছে মনে হয় যে এই সব ক্ষেত্রে আমাদের কাজ করার বিরাট সুযোগ আছে। যেমন আমি একটু উদাহরণ দেই যে আমরা এখন আমাদের যদি ডিএনএর সাথে যায় না, আমাদের কারেন্ট রিসার্চ ফোকাসড হচ্ছে হার্বাল প্লান্ট। আমাদের ওই যে ট্র্যাডিশনাল মেডিসিনাল রিসোর্স গুলো আছে সেগুলোকে ডকিং বা ড্রাগ ডিজাইনিং, এইচপিএস কে ডোজ নির্ধারণ, এনিমাল স্টাডি, হিউম্যান স্টাডি করে একটা জায়গায় নিয়ে আসে। আমরা কিন্তু এই কাজটা করতে গিয়ে বাংলাদেশের বিভিন্ন প্রাইভেট পাবলিক ইউনিভার্সিটিতে কথা বলছি, স্টার্টিং ফ্রম ঢাকা ইউনিভার্সিটি, ইস্ট ওয়েস্ট ইউনিভার্সিটি, রাজশাহী ইউনিভার্সিটি, বিসিএসআর, এবং আমরা সাংঘাতিক কোঅপারেশন পাচ্ছি। এমনকি সিলিমারিন বলে একটা গাছ যেটা পাওয়া যেত না বাংলাদেশ, আমাদের ধারণা ছিল, সেটা কিন্তু আমরা ফেসবুকে স্ট্যাটাস দেওয়া খুঁজে পাওয়া গেছে। রাজশাহী ইউনিভার্সিটি বোটানিক্যাল গার্ডেনে চাষ করা হচ্ছে, সেখান থেকে এপিআই তৈরি করে আমরা ক্লিনিক্যাল ট্রায়াল শুরু করতে যাচ্ছি। তো এই সবকিছু এটা কিন্তু কোনো ডাক্তারের পক্ষে সম্ভব না, কোনো ইউনিভার্সিটির পক্ষে সম্ভব না, এটা কম্বাইন্ড এফোর্ট লাগবে। যেমন মাইক্রোবায়োটা নিয়ে আমরা আপনার সাথে কথা বলছি যে আমরা কি আসলে কাজ করতে পারি কিনা। আমি জানি আপনারা এগ্রিকালচারিস্টরা ফার্টিলাইজারে প্রোবায়োটিক প্রয়োগ করে বোধহয় বাংলাদেশের সরকারের ফসল সার আমদানি 4,000 কোটি টাকা কমিয়ে দেওয়ার মতো ব্যবস্থা করছেন। তো এইসব ক্ষেত্রে কাজ করার অনেক সুযোগ আছে। তো আমার কাছে এই সাজেশনটা খুব ভালো মনে হয়েছে যে আমরা যদি আসলে একটা কম্বাইন্ড এফোর্ট নিতে পারি। এখানে আমি যেটা বলতে পারি যে আমরা বছর 6 আগে বাংলাদেশ স্টেম সেল অ্যান্ড রিজেনারেটিভ মেডিসিন সোসাইটি বলে একটা অর্গানাইজেশন লঞ্চ করেছিলাম, এবং সেখানে আমরা চেষ্টা করেছিলাম যে শুধু ডাক্তাররা থাকবো না, যারা বেসিক সায়েন্টিস্ট তারাও থাকবেন। তো স্টেম সেলের জায়গাটা আমরা 6 বছর পরে দেখেছি যে রিসার্চে একটা ন্যারোনেস আছে বা অত বেশি রিসার্চ হয় না। তো আমার আপনার ডিএনএ এর সঙ্গে আগ্রহটা এই ছিল যে আমরা হয়তো আগামী বছর থেকে সেই অর্গানাইজেশনটাকে আরেকটু ইলাবোরেট করে উই ক্যান হ্যাভ এ ডিএনএ মিটিং, যেখানে শুধুমাত্র স্টেম সেল নিয়ে কথা না বলে আমরা অল ব্রাঞ্চেস এর ডিএনএ নিয়ে কথা বলতে পারি, কারণ ইটস দ্য বেসিস অফ লাইফ, বেসিস অফ অল সায়েন্টিফিক রিসার্চ অ্যাকচুয়ালি। তো সেটা হয়তো করা যেতে পারে। আপনি যেটা বলছেন যে বাংলাদেশ একাডেমি অফ সায়েন্সেস ক্যান রাইটলি বি দ্য রাইট পার্টনার ফর আস। তো আমার মনে হয় যে আজকের মিটিংটা শেষ হোক, আপনি যেহেতু ইনিশিয়েটিভ নিয়েছেন আপনি লিডটা নেন। আজকে যারা এখানে একাডেমিশিয়ানরা আছে, জুনিয়রসরা আছে, তাদেরকে নিয়ে বাকিদেরকে নিয়ে উই ক্যান হ্যাভ এ ফিজিক্যাল মিটিং সামটাইম ভেরি সুন। আপনি বলে আমিও অর্গানাইজ করতে হেল্প করতে পারি। অ্যান্ড উই ক্যান থিঙ্ক অফ হ্যাভিং এ ফর্মাল প্ল্যাটফর্ম, যেখানে শুধু আমরা পাবলিকেশন করব না, ওয়ার উইল মেক রিয়েলি মিনিংফুল রিসার্চ। আপনি কি করছেন, আমি কি করছি, আমার কাছে পেশেন্ট আছে, ইফ ইউ গিভ মি দ্য প্রোডাক্ট আই ক্যান হ্যাভ এ হিউম্যান ট্রায়াল। সো বেঞ্চ বেডসের সমাধানটা আমরা করলে আমার ধারণা উই ক্যান ডু এক্সেলেন্ট ওয়ার্ক। আপনারা এগ্রিকালচারে যে কাজটা করে দেখিয়েছেন বাংলাদেশে, সেই কাজটা মানে অন্যান্য সেক্টরে রেপ্লিকেট করা সম্ভব, provided we work together. Otherwise, we will head nowhere. Thank you.
Excellent, excellent, Professor Mamun Al Mahtab. Swapnil always make something, how to say, workable. So after this meeting, one recommendation is we shall sit together and think of how we can work together. And obviously, scatteredly, we have lots of resources, if not only, intellectual capital, we have used physical capitals also underutilized in many places. And in a resource limited country, if we do not take initiative to best utilize those resources, we cannot move on. This is the reality. So one decision, I think we shall sit together just after the Eid vacation and make a platform, and DNA can unite us, because all of the researchers, either in chemistry, even physics and biology, they are connected to the DNA. Obviously, very nice comment. Thank you. Now the floor is open. Please make some other, participants, please make some comment and question. Wonderful.
স্যার আসসালামু আলাইকুম স্যার।
হ্যাঁ, ওয়ালাইকুম আসসালাম।
স্যার আমি বাংলাদেশ কৃষি বিশ্ববিদ্যালয় থেকে জেনেটিক্স প্লান্টিং এর প্রফেসর আশরাফুল হক বলছি। স্যার আপনার টকটা আমি শুনছি, ভালো লাগছে। আমার এই ভিডিওটা একটু অসুবিধা, আমি কথাই বলি স্যার আর কি। তো যেমন ডক্টর মামুন স্যার বললেন এবং আহসান হাবিব, ডক্টর আহসান হাবিব উনি বললেন যে আসলে এইরকম একটা আদলে তৈরি করা যায় কিনা, আপনিও তার সাথে হয়তো পার্সিয়ালি একমত হবেন, যাতে সায়েন্সকে আমরা কিছুটা হেল্প করতে পারি। আমাদের যতখানি যার নলেজ আছে, ক্ষুদ্র নলেজ গুলো একত্রে করে আমরা যদি একটু প্ল্যাটফর্ম তৈরি করতে পারি, আমার মনে হয় সেটা একটি ভালো আইডিয়া। তাহলে স্যার আপনি এটি একটি উদ্যোগ নেন। আমার মনে হয় যদি সুযোগ থাকে আমরা অনলাইনে মিটিং করব, করে হয়তো আপনাকে আমাদের যে সমস্ত মতামত গুলো দিতে পারলাম, অথবা ফিজিক্যালি যদি হয় সেটিও সম্ভব, আমার মনে হয় আমরা করতে পারি স্যার। এটি আমার মনে হয় একটি ভালো আইডিয়া। থ্যাংক ইউ স্যার আপনার সুন্দর উপস্থাপনার জন্য। থ্যাংক ইউ স্যার।
Excellent, Professor Ashraful. Really a great idea. And there are some scientists from different national research institute. I am expecting some comment. Hirendranath Bormon, Professor Numan Farooq, Professor Shamsur Rahman, and many others. Please make comment, and even one from Germany, Abul Khair. Mr. Abul Khair, do you have any comment?
Thanks, sir. I already wrote in the chat that.
Okay. Please share.
Yeah, I really appreciate that you and other professors from different perspectives that you are prioritizing collaborative works from home and abroad, and you are taking initiatives to create a platform where we can work from abroad as well, and we can contribute to our country with our expertise from abroad. And this is really exciting for me. And I really thanks you and organizer that you are organizing such events where we can listen your works and other people's comments, feedbacks, and we can create something from these events. That's really great, actually. And thanks, sir.
Thank you, Abul Khair. Abul Khair is a high grade researchers in bioinformatics. We are now collaborating a very fascinating project with him. He graduated from University of Göttingen, which is a home of more than 50 Nobel laureates. Really amazing, and doing a job now in a German company. Excellent. Yes, that all credit should goes to Nishat Tasnim Safa's group, because they are undergrad students and they took the this robust initiative. Thank you, Abul Khair. Any other comment?
স্যার, আমি ডক্টর হিরেন্দ্রনাথ বর্মা বলছি স্যার।
Yes, please.
স্যার, অসংখ্য ধন্যবাদ আপনার সুন্দর উপস্থাপনার জন্য। সেই সাথে আমি যারা অর্গানাইজার তাদেরকেও ধন্যবাদ জানাচ্ছি। স্যার, আমি এক কথাই বলবো যে আমাদের সব ইনস্টিটিউট, সেটা গবেষণা ধর্মী প্রতিষ্ঠান হোক বা বিশ্ববিদ্যালয় হোক, সবার একটা কোলাবোরেশন দরকার। আপনি জানেন আমাদের সব ধরনের এক্সপার্ট সব ধরনের প্রতিষ্ঠানে আছে, তো কোলাবোরেশন থাকলে, আমি যেমন ধান গবেষণায় জিনোম এডিটিং নিয়ে কাজ করতেছি এবং বায়োটেক এবং অ্যাবায়োটিক স্ট্রেস টলারেন্ট রাইস ডেভেলপমেন্টের জন্য কাজ করতেছি। আপনার বঙ্গবন্ধু কৃষি বিশ্ববিদ্যালয়ে ভালো ল্যাবরেটরিজ আছে, আপনার এখানেও জিনোম এডিটিং নিয়ে কাজ হচ্ছে। তো আমরা যদি আমাদের নলেজ গুলো শেয়ার করি, যার যেটা লেকিংস আছে সেটা আমরা গ্যাদার করতে পারবো এবং ফাইনালি আমরা একটা ভালো প্রোডাক্ট ডেভেলপ করতে পারবো। তো স্যার, কোলাবোরেশনের বিকল্প নাই, আপনি সবাইকে নিয়ে স্যার এই কাজটা করবেন। ধন্যবাদ স্যার।
Excellent. Dr. Hirendranath Burman, he is an expert in CRISPR-Cas genome editing and initiated a very nice project at Bangladesh Rice Research Institute, the senior scientific officer. Thank you so much. Very valuable comment. Obviously, without unity, we would not be able to get the independence of Bangladesh under the leadership of our father of the nation, Bangabandhu, Sheikh Mujibur Rahman. So we need, all scientists need to be united and do our best to make some impact in our society by doing science. Very nice. Any other comment?
One more comment from
Prof.
Yes. In case of the treatment of some fungus, maybe the Rice Research Institute, they are using some nanoparticles.
Yeah.
If I am right. Something like
they initiated such a project. Yeah.
Yes, silver nanoparticle or something like this. Now the question is it possible to carry out a research like a project? I want to know the presence of nanoparticles in the environment must cause the degradation or the killing of the microbes in the soil.
Yeah, this is important point. And Dr. Latif, who is newly elected a fellow of Bangladesh Academy of Science, he has initiated research on nanotechnology at Bangladesh Rice Research Institute. And in our Institute of Biotechnology and Genetic Engineering, we are also doing research on nanotechnology, but nanotoxicity and other issues, obviously we should do further work before releasing the materials. Very nice point. I have seen a raised hand from Afsara Tasnim. Yes, you can go on.
Assalamu alaikum, sir. I am Khandukar Afsara Tasnim Tutini from Agriculture 4th year, Bangabandhu Sheikh Mujibur and I am co-hosting on this program, sir.
Excellent.
Sir, I have a question for all of you, that do you think that the emphasis on research is inadequate to compared to any other sector in our country? And what do you think the reason behind this for giving less emphasis on research sector? Thank you, sir.
Yeah, thank you so much. Actually, emphasis on research, need-based research in Bangladesh, especially in agriculture, is phenomenal. If you compare with any other developing country, Bangladesh invested huge money in agricultural research and focused research. Even first, the annual budget, 20% was allocated by our father of the nation for agricultural research, and it significantly impacted. We have even a research institute for a single crop. You cannot find in other similar status of the countries. But Professor Mamun Al Mahtab and Professor Ahsan Habib, they mentioned like agriculture, as we are doing, translational research from basic to translational research, the pathway and policy decision we took in the past, other disciplines are not equally being emphasized. You are right. This is why in global mass of knowledge, our contribution is still, not significant. And there are many reasons. For basic research, investment is huge. And obviously, policymakers, their understanding of investing to generate new knowledge that are the capital for new technology. In many developing countries, it is not being realized by the policymaker. It is also our failure. We, the researchers, should come forward and convince the policymakers to, make investment and plan-wise investment for making, our knowledge capital bigger and bigger so that we can make real impact to the society. You rightly mentioned the point, and obviously we should, the researchers who are here, we should make our arguments to the policymakers, as well as if we work together, interdisciplinary approaches, as we are now here. I think all the disciplines, even social science, economics, they are here. So this type of approach has not yet been done. Bangladesh Academy of Science is a platform, but very small community, 60 elected fellows only. So we need to think of your question. It is a thought-provoking question, but it is difficult to answer in a straightforward way. I have seen Ghulam Mahbub Faisal. He raised the hand.
Yes, sir. Sir, hello, sir. I have one question, sir. I have my question to Dr. Mamun Al Mahtab, sir. Hello, sir. You are talking about the using of microbiome-based therapies, but as far I know, there is a FDA approved method, fecal microbiota transplantation. So my question is to you about if you guys are having started working on microbiome profiling. You are talking about you work with the liver cirrhosis or hepatitis patients. If you have already started working with the microbiome profiling of these patients using metagenomic methods, what would be your approach after the microbiome profiling? If you will use the probiotics, use probiotic or prebiotic. Many researchers are telling about using prebiotics instead of probiotics. So, sir, I am expecting your opinion about this. Thank you, sir.
Thank you. বাংলাদেশে এখন পর্যন্ত যে কাজ হয়েছে আমার জানা আছে সেটা হচ্ছে যে আমাদের ন্যাশনাল ইনস্টিটিউট অফ বায়োটেকনোলজি তারা সম্ভবত আমাদের যেটা হেলদি পপুলেশন এবং ট্রাইবাল পপুলেশন তাদের যে নর্মাল যে গাট মাইক্রোবায়োটা সেটা প্রোফাইলিং করেছে। তো আমরা এই মুহূর্তে যেটা করতে যাচ্ছি আমাদের ডিপার্টমেন্টে দুটো কাজ শুরু হচ্ছে। একটা শুরু হয়েছে ফ্যাটি লিভারের পেশেন্টদের গাট মাইক্রোবায়োটা দেখা, এবং একটা হচ্ছে আমার সুপারভিশনে লিভার সিরোসিসটা শুরু হতে যাচ্ছে। একটা কাজে আমরা কোলাবরেট করছি আমাদের ইউনিভার্সিটি অ্যানাটমি ডিপার্টমেন্টের সাথে, একটা ন্যাশনাল ইনস্টিটিউট অফ বায়োটেকনোলজির সাথে। আমাদের উদ্দেশ্য হচ্ছে আমরা প্রথমে দেখতে চাই যে আমরা কোনো অ্যাবনর্মাল মাইক্রোবায়োটা পাই কিনা, এবং এটা এস্টাবলিশ যে আমরা ফ্যাটি লিভার এবং লিভার সিরোসিস দুইখানে কিছু অ্যাবনর্মালিটি পাবো ফ্রম দ্য হেলদি পপুলেশন। তো এটা পেলে পরে নেক্সট কি অ্যাপ্রোচ হবে? নেক্সট অ্যাপ্রোচটা হবে যে ট্রিট করা। এখন যেটা আপনি বলছেন যে ফিকাল মাইক্রোবিয়াল ট্রান্সপ্লান্টেশন, সেটা এখন এস্টাবলিশ ট্রিটমেন্ট না, বাট আই হ্যাভ এ ড্রিম অফ ডুইং দ্যাট। বাট এগেইন দেয়ার আর টু মানে মেজর লিমিটেশনস। একটা হচ্ছে যে টু গেটিং দ্য আইডিয়াল ডোনার। এটা আমাদের যেসব কলিগে ইন্ডিয়ায় কাজ করছে তাদের সাথে আমাদের যে কথাগুলো হয়, একটা আইডিয়াল ডোনার পাওয়ার, যে স্টুলটা আইডিয়াল স্টুল হবে, ফ্রি ফ্রম অল দিস প্যাথোজেনিক ব্যাকটেরিয়া, এটা মোটামুটি ইম্পসিবল বা নিয়ার ইম্পসিবল। সো দ্যাট উইল বি এ চ্যালেঞ্জ। আর সেকেন্ড হচ্ছে যে কালচারাল শক, যে আমি স্টুল ট্রান্সপ্লান্ট করব, হাউ উইল আওয়ার পেশেন্টস অ্যাকসেপ্ট দ্যাট। তো উই হ্যাভ দ্যাট ইন মাইন্ড। আমাদের আপাতত টার্গেট হচ্ছে যে আমরা এটা করে, এবং নেক্সট স্টেজে আমরা একটা ভালো রিসার্চ পার্টনার খুঁজছি যারা আমাদেরকে একটু বলবে যে আমাদের দেশে যে অ্যাভেলেবল প্রোবায়োটিক্স গুলো পাওয়া যায়, প্লাস আমাদের দেশে প্রোবায়োটিকের সোর্সেস গুলো আছে, ফর এক্সাম্পল আচার কিংবা দই কিংবা হোয়াট এভার, সেখানে ইউজফুল প্রোবায়োটিক্স আমরা পাই কিনা। তো প্রথমত আমরা প্রোবায়োটিক দিয়ে কাজ শুরু করতে চাই, বাট আমাদের আলটিমেট টার্গেট হচ্ছে ফিকাল মাইক্রোবিয়াল ট্রান্সপ্লান্টেশন। বাট এই লাইনে আমরা একদম আসলে নতুন, উই হ্যাভ জাস্ট স্টার্টেড। বেসিক্যালি আমার রিসার্চ ফোকাস ওয়াজ লং হেপাটাইটিস বি, এবং আমরা এটা ড্রাগ ডেভেলপমেন্টের সাথে রিলেটেড ছিলাম। কিন্তু অনেকদিন কাজ করার পর আমাদের যেটা উপলব্ধি সেটা হচ্ছে যে যদি বেসিক সায়েন্সটা আমাদের কন্ট্রোলে না থাকে, তাহলে আমরা অনেক পেপার করতে পারবো, কিন্তু আমাদের পপুলেশন বেনিফিটটা পাবে না। ফর এক্সাম্পল, আমি ন্যাসড্যাক বলে একটা, আমার যে রিসার্চ গুরু বলতে পারেন, তার নাম হচ্ছে ডক্টর ফজলে আকবর, ইজ দ্য জাপানিজ বাংলাদেশি। তো উনি আর আমি মিলে কিন্তু দ্য ড্রাগ ইজ হিজ কনসেপ্ট, ইট ওয়াজ ডেভেলপড বাই দ্য কিউবানস, বিকজ এটা একটা যে বায়োটেক প্রোডাক্ট। সাবসিকুয়েন্টলি ফেজ 1, ফেজ 2, ফেজ 3 ক্লিনিক্যাল ট্রায়াল সব বাংলাদেশে আমার করা। ওষুধটা কিন্তু এখন কমার্শিয়ালাইজড হচ্ছে কিউবা সহ পৃথিবীর 5টা দেশে। বাংলাদেশে কিন্তু 2 বছর ধরে আমরা কোভিড আগে থেকে চেষ্টা করছি, আমরা এটা অ্যাপ্রুভালও পেয়েছি। দ্য ফার্স্ট ড্রাগ টু বি ডেভেলপড ইন বাংলাদেশ, যেটা বাংলাদেশে আমাদের ড্রাগ কন্ট্রোল কমিটি অনুমোদন দিয়েছে। তো ওয়াই ইজ দ্য ড্রাগ নট কামিং হিয়ার? এটা হচ্ছে যে এখানে যেটা দেখতে পাচ্ছি যে ইটস এ কমার্শিয়াল গেম। ফার্স্টলি পেটেন্ট আমাদের না, পেটেন্ট ওয়াজ টেকেন অ্যাওয়ে বাই দ্য কিউবাস, আমরা পেটেন্ট বুঝতামই না। রাইট নাও উই আর নেগোশিয়েটিং, এটা একটা অন্য পেটেন্ট হয়তো আমরা এখন ফাইল করতে যাচ্ছি। সেকেন্ড হচ্ছে যে ড্রাগ কোম্পানিজ প্লে দ্য রোল, মানে এখন আমার দেশের ড্রাগ কোম্পানি প্রফিট হিসাব করে চিন্তা করছে যে ন্যাসড্যাক আনবে কি আনবে না। সো হিয়ার সায়েন্স বিকাম ভেরি আনইউজফুল অ্যান্ড লেস ইউজফুল। তো সেই জন্য এখন তো নাও আই এম 52, I don't think I have very productive, many years of productive life left. না, আমার এখন যেটা রিসার্চ ফোকাস হচ্ছে যে টু ইউজ দ্য লোকাল রিসোর্সেস দ্যাট উই হ্যাভ। সেজন্যই কিন্তু আমরা এখন হার্বাল প্রোডাক্টস নিয়ে কাজ করতে চেষ্টা করছি। Right now we are working with 5-6 products like amloki, like orjun, like moringa, omega-3 fatty acid, silymarin, কালোমেঘ বা গ্রিন চিরতা। এই প্রত্যেকটা কিন্তু আমরা ডকিং, HPLC, অ্যানিমাল স্টাডি, হিউম্যান স্টাডি এভাবে প্ল্যান করে আগাচ্ছি, এবং 2টা অ্যানিমাল হিউম্যান স্টাডি শুরু হয়েছে। So there are challenges. Challenge হচ্ছে যে you just cannot do what you want. So microbiota is very close to my heart, because এটা আমাদের দেশে আমাদের পক্ষে করা সম্ভব, এবং we can take a stage forward. তো এখানে তো অনেক সায়েন্টিস্ট আছেন, জাস্ট ফর মাই নলেজ আমি এটা তোফাজ্জল ভাই আপনার পরামর্শের প্রশ্ন করতে চাই। আমরা তো কাজ করছি, বাট অ্যাক্টিভ ইনগ্রেডিয়েন্ট আইসোলেশন করার মতো টেকনিক কি আমাদের এখানে আছে নাকি? ফর এক্সাম্পল, আমি যখন গ্রিন চিরতা বা কালো মেঘ নিয়ে কাজ করছি, আমি দেখছি 23টা ইনগ্রেডিয়েন্ট আছে, যার মধ্যে অ্যান্ড্রোগ্রাফিলিডি হচ্ছে দ্য ইউজফুল ইনগ্রেডিয়েন্ট দ্যাট ইজ পসিবলি হেল্পফুল ফর ফ্যাটি লিভার। একটা আমরা ডকিং করে দেখেছি এটা 5টা জায়গায় কাজ করতে পারে। আই ক্যানট আইসোলেট অ্যান্ড্রোগ্রাফিলিডি। এটা কি সম্ভব কিনা কোনো একটা ল্যাবে আমাদের যদি এই তথ্যটা আমাকে দিতে পারেন, তাহলে কিন্তু আমার কাছে মনে হয় যে আমাদের হার্বাল মেডিসিনের রিভাইভাল আমাদের দ্বারাই সম্ভব।
থ্যাংক ইউ সো মাচ। আমার মনে হয় যে প্রফেসর মামুন আল মাহতাব খুব চমৎকার কথা বলেছে যে রিয়েলিটিটা কি এবং আমরা কিভাবে এগোতে পারি। তবে আপনি যে গোলাম মাহবুব ফয়সাল খুব ভালো প্রশ্ন করেছে, আমাদের ভেটেরিনারি সায়েন্সের স্টুডেন্ট। আমি স্বপ্নিলের এই প্রশ্নের একটা উত্তরে বলি, ইয়েস, বাংলাদেশে আপনি যদি হার্বাল মেডিসিনাল প্রোডাক্টের ক্যারেক্টারাইজেশন করতে চান, এটা সম্ভব। আমি আমার লাইফে 56 নিউ ন্যাচারাল প্রোডাক্ট মোস্টলি প্ল্যান্ট এবং মাইক্রো অর্গানিজম থেকে ডিসকভার করেছি। এখানে বাংলাদেশে একটা বড় লিমিটেশন ছিল আমাদের, যেটা হচ্ছে এনএমআর এবং এই স্পেকট্রোস্কোপিক মেথড। এখন HPLC হয়েছে এবং এনএমআর এবং আদার মেথডের ক্ষেত্রে ডাটাবেস তৈরি হয়েছে। প্রফেসর হার্টমুটলা আছে, আমার এক সময় কোলাবোরেটর ছিল, সে একটা ফ্যান্টাস্টিক ডাটাবেস করেছে। কাজে সিম্পল এনএমআর এবং এইচপিএলসি দিয়ে পিউরিফাই করে আমরা কিন্তু যদি নোন কম্পাউন্ড হয় ইনস্ট্যান্টলি স্ট্রাকচার পেয়ে যেতে পারি। আর যদি আননোন কম্পাউন্ড হয় তখন আমাদের খুব ভালো কোলাবোরেটর আছে ইন্টারন্যাশনালি, যাদের কাছ থেকে হয়তো 2D এনএমআর ডাটা বা এরকম আনতে হতে পারে। কিন্তু এখন বাংলাদেশে এনএমআর মেশিনও আছে। এনএমআর ডাটা আমরা বিসিএসআইআর থেকে নিতে পারি, জাহাঙ্গীরনগর ইউনিভার্সিটি থেকে নিতে পারি। মূল বিষয়টা হচ্ছে কি, রাইট লুক গুলোকে এমপ্লয় করা। আমি একজনকে জানি, ডক্টর মজিদ মন্ডল, আমি মনে হয় সে পার্টিসিপেট এখানে করে নাই, কোরিয়াতে একেবারে ট্রুলি এই আপনার ডিসকভারি অফ নিউ ড্রাগ, সে অরিজিনালি ফার্মেসির ব্যাকগ্রাউন্ড, রাজশাহী ইউনিভার্সিটির ফার্স্ট ক্লাস ফার্স্ট ছিল, সে লটস অফ নিউ কম্পাউন্ড ডিসকভার করেছে এবং জার্মানিতে কাজ করেছে হুম্বল ফেলো হিসাবে। তো এখন দেশে ব্যাক করেছে, আমি তার ল্যাবও ভিজিট করেছি। এইচপিএলসি সহ ও খুব ফাস্ট আপনার মলিকুল পিউরিফাই করে। আর এই আমরা হয়তো প্রয়োজনে দেশের বাইরে যদি 2D এনএমআর এর প্রয়োজন হয় নিব, আর দেশে যদি ওইভাবে একটা আমরা একটা এই ইয়া করি, ইন্টিগ্রেটেড টিম করি, কার পরে কে কাজটা করবে রিলে, তখন খুব দুর্দান্ত হবে। এবং এটা আমি মনে করি আপনি যদি লিড নেন, I can assemble the right people, এবং এটা একটা বড় রকমের কাজ হবে। আমরা যদি স্পেসিফিক অ্যাক্টিভ ইনগ্রেডিয়েন্ট, তাদের বায়োলজিক্যাল অ্যাক্টিভিটি, সেফটি সবগুলো কনফার্ম করে আমরা রিয়েল লাইফ অ্যাপ্লিকেশনে যেতে পারি, যেটা আপনার এখানে ফ্যাসিলিটিস আছে ক্লিনিক্যাল বিষয়, এটা দুর্দান্ত। আরেকটা যেটা মাইক্রোবায়োমের কথা আপনি বলেছেন, মাইক্রোবায়োমে আমরাও কিন্তু খুব ইন্টারেস্টেড, কারণ আমাদের ইলুমিনা সিকোয়েন্সিং যে প্ল্যাটফর্মটা আছে, এটা দিয়ে আমরা মাইক্রোবায়োমের থরো স্টাডি করতে পারি। এবং আমাদের গ্রুপে এখন আপনার মেটাজেনোমিক স্টাডি ওয়ার্ল্ড ক্লাস আপনার এই এক্সপার্ট আছে। কাজে এইটাও যদি ওই রিলে টাইপের টিম যদি করা হয়, খুব ফাংশনাল টিম, তখন খুব দ্রুত আপনি এগুনো যাবে। আমরা তো শুধু একাডেমিক রিসার্চ নিয়ে এখানে চিন্তা করছি না, রিয়েল লাইফ অ্যাপ্লিকেশন। আমার রিসার্চ কিন্তু আপনি একাডেমিক পাবলিকেশন হচ্ছে, বাট মূল ফোকাসটা হচ্ছে টু সলভ দ্য প্রবলেম এবং নিডটা কি। ওখানে পাবলিকেশন করতে তো আপনার কোনো অসুবিধা নেই, ইভেন প্যাটেন্ট করতে অসুবিধা নেই। সেই জায়গাগুলোতে আমাদেরকে যদি আমরা রাইট লুক গুলোকে এনগেজ করে ওইরকম কনসোর্টিয়ামের মতো কাজ করতে পারি, দ্যাট উড বি হিউজলি ইম্প্যাক্টফুল। এবং আপনি এখানে কিছু লিডারশিপ দিবেন, আমার দিক থেকে ফুল সাপোর্ট আপনার সাথে পার্টিসিপেট করব। থ্যাংক ইউ সো মাচ।
আমি একটু খালি আমি আপনার 100% একমত যে আমরা যে স্ক্যাটার্ড রিসার্চ গুলো করছি, আমরা ওয়ার্ল্ড রিসার্চ লিটারেচারকে সমৃদ্ধ করব, দ্যাট ইজ নট ইজ হোয়াট ইজ এক্সপেক্টেড ফ্রম আস। এবং আমাদের জেনারেশন আমরা সায়েন্সে বাংলাদেশের নোবেল লরেট পাবো, সেটাও মনে হয় সম্ভব না। যেটা আপনারা এগ্রিকালচারিস্টরা করছেন, মানে সায়েন্স দ্যাট ইজ ভিজিবল, চালের উৎপাদন বাড়ছে, গমের উৎপাদন বাড়ছে, দ্যাট ইজ পারহ্যাপস ডুয়েবল ইন মেডিকেল সায়েন্স অলসো ইফ আই কোলাবরেট উইথ ইউ। মানে আমরা যদি আপনার সাথে যোগাযোগ স্থাপন করতে পারি, দেন দ্যাট ইজ ডুয়েবল। আমার মনে হয় আমরা যেটা বসার কথা হচ্ছে, সেটা আসলে বসতে হবে এবং এইগুলো একটু করতে হবে, কারণ অনেকেই ইন্টারেস্টেড আছেন। এবং আমাদের যেটা বড় স্ট্রেংথ, আমাদের তো এমডি বা পিএইচডি স্টুডেন্টরা থাকে, তো তাদের কিন্তু একটা ক্লিনিক্যাল রিসার্চ করতেই হয়। তো আমার আন্ডারে যারা আসে বছর 7-8 জন, তাদের কিন্তু এখন কম্পালসরি ক্লিনিক্যাল ট্রায়াল করতে হবে। সো উই হ্যাভ দ্যাট রিসোর্স এন্ড উই নিড টু ব্যাক আস আপ।
এই আইডিয়াটা আমাদের আগে হয় নাই। যেমন মনে করেন কেউ মেডিকেল রিসার্চ করছে, কেউ এগ্রিকালচার রিসার্চ করছে, কেউ বুয়েটে রিসার্চ করছে, কেউ ঢাকা ইউনিভার্সিটিতে রিসার্চ করছে। এই যে প্রত্যেকের স্ট্রেংথ গুলো আছে, এগুলো কিন্তু ডিসক্রিট, তাদেরকে ইউনাইট করলে আমরা রিয়েল প্রবলেমটাকে মানে পারফেক্টলি সলিউশন করতে পারবো। এবং এই জায়গাটাতে যাওয়ার এখন আমার মনে হয় আমাদের মাইন্ডসেটটা এসেছে। আগে কিন্তু এইভাবে যার যার মতো করে যার যার অ্যাসোসিয়েশনে লোকজন থাকতো, এই জায়গাটা এখন আমি মনে করি বাংলাদেশে এই আবহাওয়াটাও তৈরি হয়েছে। এক্সেলেন্ট। এবং আমি রেডি আছি, এবং আমার মনে হয় অনেকে এখানে যারা জয়েন করেছে তারাও এরকম ইম্প্যাক্ট করতে চাই রিয়েল লাইফে। পাবলিকেশন করে গ্লোবাল মাস অফ নলেজে শুধু সাইটেশন, এইটা আমাদের এখন গোল হওয়া উচিত না। এটা অটোমেটিক হবে আপনি যদি ইম্প্যাক্টফুল ওয়ার্ক করেন। যেমন আমরা হুইট ব্লাস্টের ক্ষেত্রে যেটা করেছি, গ্লোবাল যত সায়েন্টিফিক কমিউনিটি আছে, যে যেখানে পাওয়ারফুল আছে, প্রবলেমটা আমি তার সলিউশনের জন্য লোকজনকে এনগেজ করছি, এবং ইট ইজ ওয়ার্কিং। ইনক্রেডিবল, আপনি অবাক হয়ে যাবেন। তারা সবাই কিন্তু এগ্রিকালচারের লোক না, তারা বিভিন্ন ডিসিপ্লিনের লোক, কেউ ইভেন আপনার ইভলিউশনারি বায়োলজির লোক, কেউ মেডিকেল সায়েন্সের লোক, কিন্তু এক একজনের এক একটা স্কিল আছে। ওই স্কিল গুলো একত্র করলে একটা পাজল সলভ করা, যেরকম আপনি একা করলে 1 মাস লাগবে, কিন্তু আপনি 7 দিনে করতে পারেন যদি 7 জন আসে একসাথে বসে। এটা সম্ভব। থ্যাংক ইউ সো মাচ। এটা আমার মনে হয় খুব গ্রেট আমাদের ফোকাসের জায়গায় আমরা আসছি। আর কে প্রশ্ন করবে? রঞ্জন সিং। আমার মনে হয় অনেক লম্বা আলোচনা হয়েছে, আর 5 মিনিটের ভিতরে আমরা শেষ করতে চাই আলোচনা।
Hello, sir. Greetings. My question is that earlier you talked about Bermuda Conference. And
would you please introduce yourself, from where you are participating?
I'm Rajan Singh. I'm a Nepali student in agriculture 4th year.
Excellent, excellent. Yeah.
My question is, earlier you talked about Bermuda Conference, and it's about making the Genome project accessible to public in 24 hours. Now with the arrival of powerful AI tools, what are the chances of misuse of those data? Because recently when I doing assignment on bioinformatics topics, many scientists and researchers have raised problems on misuse of data as AI is becoming more powerful, and recently Chinese scientists also broke the scientific norms and ethics by.
Yes, I got your point. For example, when CRISPR-Cas methods, the sharpest genome editing tool, was discovered, Chinese scientist Jian Kui, he edited the germline of human, and which was seriously protested by the global scientific community. Why? Because the method is not yet been, optimized and 100% precision for application in the human. But it happened, and it stopped due to global protest. So global scientific community should maintain some ethical guidelines, and every country should follow it. And in case of NCBI or other gene banks, every second thousands of sequencing data and information are deposited over there. Due to artificial intelligence, emergence of artificial intelligence, some people may abuse them. It is true. But, to consider the bigger issue, there are some trade-off. But we need to have the laws and other regulations to protect and restrict the people to abuse the opportunities. But there is a chance, you are right. In every aspect, if science is utilized for to harm the human, the science which is sharpest, the chance of getting more danger is possible. For example, Twin Tower was damaged by 2 aeroplanes. Aeroplane is not was not made for hitting the Twin Tower or like that, but it may happen anyway. This is the important point. The biosafety, bioethics, and other guidelines is necessary. I have seen Aparna Islam, who is our distinguished panelist. She raised the hand, and obviously she can make some more points. And we are nearly at the end of our journey, but we are very happy that Professor Aparna managed time to join with us. We had 144 participants, really a great and grand celebration of the World DNA Day. Now, Professor Aparna Islam from BRAC University, she has lots of expertise in biosafety as well as the biotechnology in plant and microorganisms. Professor Aparna.
Assalamu alaikum shabaike. I'm very sorry. I have joined only 10 minutes back. You would be very happy to know that International Society for Biosafety Symposium, ISBR, this is the 16th ISBR Symposium, which is going to be held on 30th of this month in St. Louis, USA. And we were at the final stage of preparation, and I was stuck into that meeting. And from Bangladesh, couple of eminent scientists, researchers, regulators, they are joining the symposium. I think in total it will be around 20 people who will be joining. And I like the idea of getting consortium-based research. And I think Professor Tofazzal Islam is the pioneer of putting up a consortium which is effective, not only among the scientists of Bangladesh, but also scientists from abroad. He brought it together and then made a powerful research outcome. And now in Bangladesh, this has actually initiated a culture of consortium. And like I'm in a university, I'm working with the industry where they have an aim for agricultural target oriented research, where we are collaborating with them. And regarding the instrument that Professor Islam have just said, we have HPLC. A lot of HPLC is actually there. NMR is there. Even RT-PCR facilities are now very much available in the universities and the research institutes. So it's just that finding out the key person and doing a collaboration. One experiment or one portion of the research can be done in collaboration with that. And regarding, I'm just talking about what I have heard regarding collaboration among different disciplines. Like I have a research field where my EEE faculty are using it for their AI robots for understanding how good their robots are to have the data from the far away. So the farmer doesn't have to go to the field every day. They can send the robot and get the thing. But the last question, which was raised, bioethics, ethical issues, this is very important. We scientists have huge power in our hand because knowledge is so open nowadays. But with power comes the responsibility. It's our responsibility. And if we forget to exercise our responsible nature, then of course there are rules and regulation. As I was saying in the beginning, that biosafety symposium is there, and I work with the biosafety. Professor Islam is also there. So as a biotechnologist, we need to know what is biosafety, what are the rules and regulations that our country have, and what are the rules and regulations other countries internationally there are, and we have to follow it. It's not for the own ethical question only, but also if I don't produce a product which is safe, then I cannot expect my product to be consumed by my consumers or to be used by my audience or the end user. The basic point that we have to think that whether my research is getting translated successfully so that it is being used by my people. Only publication is important. Publication actually makes me visible in the world arena that I am expert in that or I'm working in this field. But getting my product into the field, in my case, it is into the field or into the market, is very important, very important. And to get that trust that I'm doing a good science and making a good product, we have to be within the regulatory system. We have to follow it. And as a biotechnologist, I am obliged to do it. So we have to remember that. The DNA Day started with the Human Genome Project when I was doing my PhD. In Nature, there was an option where if you write to them regarding what you are doing, and then you ask for the CD with the whole genome project sequence, they would have sent it to you. During that time, we have to write it down, ask for it. At this moment, NCBI is open to all, and we can just get it. So information is there. Now it's our turn to use it responsibly. So I thank Professor Tofazzal Islam for arranging this thing, and I feel so sorry that I couldn't attend it earlier. But I'm sure it was a very fantastic program with lots of participants and lots of questions, because it started at 8 and it is almost 2 hours now. So thank you for calling me, and I'm so sorry about not being present here. Thank you.
Yes, in every meeting, she is, the first person appear in the meeting. But today, there was reasonable, background why she is a bit late, but late better than never. And really, at the end, you made very nice and insightful comments, and thank you very much. And we are at the end of our journey. As one student mentioned, opening the data, open science is maybe problematic for artificial intelligence and others. You see, look, our recent past, that is for fighting against the SARS-CoV-2 virus and GIS AID every day. Hundreds of sequence data were deposited that help us to understand the variants, all the variants, and design the vaccine. So we have to focus the benefit, and obviously, as Professor Aparna mentioned, ethical issues are becoming more powerful. Not only biotechnology, even IT, other digital technologies. Many people are abusing. So we have to have new, regulatory mechanism, smart regulatory mechanism to stop those evil activities. Anyway, this is the way we have to move on. Last question, Khalid, do you like to ask question? You raised your hand.
Yes, sir.
Okay, your question would be the last question because I didn't see any other hand raised.
Okay, sir.
And meanwhile, Nisha, please go on your last program. That is, do you like to announce the result of the competition?
Sir, there is a quiz session.
Quiz session, is it going on?
Yes, sir.
Okay. And how about your
permission to open the quiz form, sir?
Huh?
Sir, we expected your permission to open that quiz form, sir, in our chat list.
Yeah, please open the quiz form.
Thank you so much, sir.
Okay, and also we can announce the other competition. So you can announce and we can listen and clap. And obviously, I promise to offer the attractive gifts to all the winners, and even all the participants will receive a fantastic certificate signed by a panelist and the keynote speaker.
Yes, sir. The result is already in our hands, sir.
Great. So Khalid, please make a very brief question, and then we shall go for the result announcement.
Okay, thank you, sir. Firstly, I would like to thank you for writing a book, one genome editing in Bangla, in this last book fair. So it ignites me to think about DNA and also genome. So I have a question here. It's a little bit interesting question. Is it possible to make whole genome sequence by artificial intelligence by just watching a photo of a man? Sir, please, if you answer this.
Yeah, at this phase, I saw what YouTube post by taking the image. Artificial intelligence is giving the genome sequence of human, but the error rate is very high. But one day maybe the artificial intelligence, the way is fertilizing the cross-fertilizing the biology. One day it may happen, but it is still premature to have the whole genome sequence by simply image analysis by the artificially intelligent software. This is the current status, but in future what would happen? Difficult to say. 10 years, coming 10 years, what would happen? It is impossible to say, because in the past, especially the mathematics, computer science, they had little interest about biology. They had interest in other things. Now, algorithms are frequently, and they are mostly focusing the biology, and biological data also generating so fastly, not only genomics data, proteomics, all other omics data. So, which is difficult to handle by, our common sense as well as our common software. So big data, machine learning, and ultimately artificial intelligence would significantly impact on biology and understanding of the biology. Mamun Al Mahtab is here. Maybe in future he will ask when we visit him, please give your DNA sequence data, genome sequence data. We'll go with a memory card or like that. Even we can have a barcode in the global gene bank, just simply and get the profile or like that, and it would happen surely, and would move. The coming days would be more fascinating. Those who are younger than me, you can see more fascinating and exciting thing will happen, and we are expecting you will participate and be a player on those changes and make the change for the society. Thank you so much. Mahbub Rabbani, do you like to make a comment? Our General Secretary of Bangladesh Nano Society, very brief, and then I shall move to Afsara Tasnim Totini.
Okay, thank you so much, sir. Assalamu alaikum and welcome everyone. So I know your arrangement from the beginning, and I'm really astonished to see still 77 participants are listening the program. In this time, people don't want to listen any webinar, but this number significantly indicates us that people are interested to learn about DNA. So I'm really happy to see your program and participants and interests of the participants. Thank you very much, and I hope this program will be arranged again and again every year. Happy
the General Secretary of Bangladesh Nano Society, DNA is a natural nanoparticle. So everyone has DNA.
Yes.
It is difficult to leave the DNA, impossible. So now I'm closing the discussion session with profound thanks to all the participants, all the organizers, as well as the discussants. Really amazing evening. We spent more than 2 hours, and Dr. Mamun Rabbani really said, in case of our university program, even national program, even our academic program, we cannot have 150 participants and a 2 hours long discussion. 77 still remain is really a big record, and we made the record on this special day, World DNA Day. Thank you so much. Now I'm requesting the organizers to declare the winners of the competition, including the quiz, if you have the results. So my job is over, and I am really thankful. And I again promise all of you that everyone will receive the participation certificate, as well as all winners will receive attractive, prizes from me. Full responsibility. Thank you.
Thank you so much, sir, for conducting the most informative and your cordial question and answer session. Thank you so much, sir. We really learned a lot, sir. Dear audience, we have come to the most exciting part of our program, the prize giving ceremony. A large number of students from all over the country participated in the competition organized at DNA Fest, and we selected the winners with very close margin of marks on the basis of mass evaluation, post-like comments by our expert judges. Our competition had 3 segments: article writing, poem or storytelling, and digital content presentation. And the results are already in my hand, and soon I'm announcing that. At first, I will announce the result from the segment poster. The second runner-up took place Nasimuzzaman Sabbir. First runner-up Shaheen Iftikhar Ritik and Rudronil jointly. And first run and champion take place in poster segment Jinia Islam Ima and Samia Shama. Congratulations all of you to take place as winner. Now I will declare the winners for the segment article writing. Second runner-up took place combinedly Sumaya Yasmin, Tonima Islam Tonu, and Rakib Hassan Mubin. All of them got same mark simultaneously. First runner-up took place Tanjuma Tabassum Tithi, and the champion of the segment article writing took place combinedly Mohammad Yunus Kobir and Rudronil Monto. Congratulations all of you. And we have come to the last section of our competition, and it was poem writing. Second runner-up took place combinedly Samiha Mubashira and Tanjim Tahir. First runner-up took place Nitu Shikdar Dipto, and it's really a matter of joy and interest that champion in this segment took place both of the hosts of this program, Nisha Tasnim Safa and me, Khondokar Afsara Tasnim Tutin.
আর্টিকেল ফার্স্ট কি বললেন?
In article writing, first place took place Mohammad Yunus Kobir and Rudronil Mondol. This species, sir,
have you got the results of quiz?
Sir, if you tell something for the winners and the participants.
Simply our expression from all the participants here, we express our profound thanks, congratulations to the participants, because within a very short notice, you participated the event and spent your intelligence and creativity. I visited the DNA Fest 2023 page, and all of the participants, please visit that page and find all the posters, poems, as well as the essay. They really did a fantastic job. I didn't thought my students are so powerful in their imagination and so brilliant. Really very, very exciting moment. And please share all the, participants, the activity that is poster, poem, and all others. Please share with all. We have the provision to, we have the provision to award only first, second, third, but I think all of you received equally our heartiest congratulations for your participation. So participants should get a separate certificate of participation in the competition. So please give all the participants, those who competed in the event, a special certificate. And I hope all the organizers who organized this event voluntarily, they should get a special certificate from us. So
thank you so much.
This is the way we can, cherish ourselves and be happy. And happiness is very important. I think the meaning of life is to become happy. And today I am very happy to see such a nice, organization of the event and active participation. So last quiz competition, do you have the results now? Afsara, Tasneem?
Sir, soon we will get the result from quiz test.
It takes time. Is it taking time or?
Sir, it may take time, sir.
Okay, then you can announce later on. So
Yes, sir, obviously, sir.
As organizer, Nishat Tasneem and Afsara Tasneem, both Tasneem. Very nice.
Yes.
Please close the session with your, voices about the current program, your experiences, as well as what you are expecting in future. So the closing voices from Nishat Tasneem and Afsara Tasneem. And I finally express my sincere thanks to Bangladesh Agricultural Olympiad, who spontaneously outreach this program and help the student to make this event very successful. Thank you.
Thank you so much, sir. And best wishes to all the participants for their participation and enthusiasm. And congratulations to the winner. Now, Nishat Tasneem Safa, I am giving you the floor to finish this program.
Thank you so much for giving me floor. I would like to extend my heartfelt gratitude to Professor Dr. Tofazzal Islam sir, our esteemed keynote speaker, and our esteemed panelists, and all of our guests for joining us in celebrating World DNA Day. Let us continue to explore the mysteries of DNA and advance our understanding of life's fundamental building blocks. Thank you all for being a part of this momentous occasion, and let us embark on this exciting journey of discovery together. Thanks to all.
Thank you so much. Now I am closing the program. And obviously you can see the video record through YouTube. We shall share hopefully if it is recorded correctly. Thank you once again, and good night to all of you.